So today I'm going to present a study, a preclinical study in a sheep posterolateral fusion model in which we compared 4 different bone substitutes. My declaration of interest: I am an employee of Kuros Biosciences. So to start, I want to talk about a bone graft substitute which has a submicron surface topography. It's a biphasic calcium phosphate that is highly porous, and it is embedded in a fibrillar bovine collagen matrix. And this has a surface topography, which is also shown on the scanning electron microscopy images of needle-shaped crystals that are smaller than 1 micrometer in diameter. And through preclinical research, we have discovered that this surface topography has an effect on the immune response of macrophages, so our white blood cells, and it polarizes them into the pro-regenerative M2 macrophage subtype, which is able to communicate with stem cells and lead to osteogenic differentiation. To study this, we cultured macrophages on the surface of this structured calcium phosphate and compared it to conventional calcium phosphates. And when we tested this material in a more basic science model, a soft tissue implantation, so we implanted this material in a muscle pouch of a larger animal model, and then we extracted these samples again and performed histology. We discovered that it's able to form bone, so it's an osteoinductive calcium phosphate, whereas in general ceramics like calcium phosphate are only osteoconductive. So in the current study we were evaluating this specific material, osteoinductive biphasic calcium phosphate, to 3 other synthetic bone substitutes. The first being an anorganic bovine bone matrix with a 15 amino acid peptide putty. The second being a nanosynthetic silicate-substituted calcium phosphate putty. And the third being a micro and nanofiber-based bioactive glass collagen matrix. So in the table below we see how these materials were used. So both of the collagen matrices were embedded with BMA, so bone marrow aspirate, in a 1:1 ratio. And then all of these grafts were implanted standalone, so not mixed with any autograft, in the posterolateral spine of the sheep at 3 levels. And 10 cc of graft was implanted for each treatment. So after 12 weeks, a range of motion testing was performed on the spinal explants to measure the range of motion in the treated segments. And the results indicated that the silicated calcium phosphate and the bioglass group had a much higher range of motion in lateral bending. And the silicated calcium phosphate group also had a much higher range of motion in the flexion extension group. Whereas the osteoinductive calcium phosphate had a lower range of motion than a previous autograft control group from a similar model. Here we see radiographs from the implantation. So we see the BCP, the bioglass, the peptide-based bone graft, and the silicated calcium phosphate. We can see the graft material between the transversal processes. What is notable here is that in the peptide putty group we see a lot of migration of the graft away from the implantation site. Next to the radiographs we have micro CT samples, so these are very fine detail reconstructions of micro CT scans, and we have a beautiful image of the fusion masses, which look fairly consolidated for the osteoinductive calcium phosphate. And also for the other groups, there appears to be continuous fusion between the transversal processes. However, when we look at the histology, we can actually observe that if we look at the detailed tissue composition, there's a lot of fibrous tissue present in the bioglass group, in the silicated calcium phosphate group, and the peptide-based putty group. Whereas the osteoinductive calcium phosphate is grown full with mature lamellar bone with bone marrow spaces. And based on these histological images, we determined the fusion rate. And as you can see in the table at the top was 100% fusion for the osteoinductive calcium phosphate, and then the putty and bioglass based had a 0% fusion rate, and the silicated calcium phosphate had a 50% fusion rate. So conclusions across multiple assessments in this clinically relevant preclinical PLF model, we determined that this osteoinductive calcium phosphate achieved superior fusion rates as a standalone bone graft compared to the 3 competitors. It exhibited robust and mature new bone formation, whereas the others indicated graft migration or lack of bone formation in the soft tissues between the transversal processes. And yeah, we observed migration of the peptide-based putty in 2 out of 3 implants. Thank you very much.
Thank you.